Okay, so I'm going to start today with a picture that's really familiar to most of us, but I think it's worth looking at again just to sort of appreciate the sheer scale of the problem that we're facing in terms of genetic variation data. So we've kind of moved from having no human genome sequenced to having millions of human genomes sequenced in a very, very short amount of time. And I think this is driven by three principal factors. The first factor is cost. It's gone from costing billions of dollars to sequence a single genome back in the early 2000s to costing $1,000 now. So we're sequencing humans because we can. We're also sequencing humans and other organisms because we've learned a huge amount about biology from doing this. There's massive insights being gained into all sorts of different fields. And the third factor that's driven this explosion in human genomes that have been sequenced is the medical applications. We've got real medical insights into diseases from doing this. So it seems likely that there'll be continued investment in sequencing human genomes, and we're going to have a very, very large number of samples in the not too distant future at all. So just to be clear, the type of data that I'm talking about today isn't the sort of raw reads that we sequence. It's the data that we get at the end of a very complex and costly pipeline to turn those reads into variation data. So we've got 3 billion bases roughly, and there's no point in writing out those 3 billion bases for every human that we sequence. We just store the bits that are different from the reference. But even by just storing the bits that are different from the reference, we still have millions of variants. So every sample has millions of data points associated with it. And the problem, I think, is that the methods and the algorithms and the data structures that we have for dealing with these huge numbers of, or dealing with variation data are lagging significantly behind our ability to produce variation data. And I think it's fair to say that the majority of the software that we have and the majority of the algorithms underlying the software will struggle with 100,000 samples and more. And this is perfectly understandable. We've gone from having sample sizes of 1,000 to sample sizes of roughly 1 million in about 5 years, which is an astonishing growth. And I think the underlying pattern there is that, well, the algorithms that we had in the sort of 1,000 genomes era, you could have quadratic time algorithms when you got 1,000 samples. You know, if you spend enough time shaving constants off of your quadratic time algorithm, you put a lot of engineering effort into it, then you can make it work. It is feasible. But if you have 1 million samples, then there is no quadratic algorithm that will work. You can't throw hardware at this, and you can't use fancy computing instructions. It'll still never be feasible. So I would argue that we need a new class of algorithm and a new class of data structure to work with these very, very large sample sizes. So a possible solution that I would like to propose is to use trees. So what our compression methods and our analysis algorithms spend an awful lot of their time doing is finding shared structure in the data. And a lot of the time, this shared structure is a direct consequence of the family tree structure of the data itself, how our samples cluster together into their ancestors. So the claim that I want to make, the central claim of the talk, is that if we can use the underlying tree structure of the samples, then we can enable both very, very efficient compression and have very efficient algorithms for processing the data. So the talk is about a data structure. And the data structure we call the tree sequence is motivated directly by coalescent theory. And the tree sequence is efficient precisely because it models the underlying processes that generate the genetic variation that we observe today. Coalescent theory also provides us some really useful analytical tools to help us analyze the performance so we can do real computer science on this problem. So what we're doing is we're using the mathematical results of population genetics to help us tackle a very, very difficult computer science problem. So what we're going to do is show that the tree sequence data structure, it hugely increases the scope of what can be simulated. It's very, very efficient to process, and it potentially provides us a method that we can use to efficiently store and analyze observed genetic variation data. So we're going to have three basic sections to the talk. The first section, we're going to talk about simulating and storing genealogies. The second part, we're going to talk about how we use these genealogies to encode variation data. And then the third, much shorter section of the talk, we're going to talk about ongoing work on how we estimate these genealogies from observed data. So the first part, we're simulating and storing genealogies. I'm just going to put my cover down here. So this is all built on top of coalescent theory. And coalescent theory is a very important theory in modern population genetics. And its sort of defining feature is that it takes a retrospective view of population processes. So given our samples, S1 to Sn, we think about how their ancestors relate to each other. And the coalescent, the sort of core idea of the coalescent is a genealogical tree. So we've got an example of a coalescent tree here. So we have our samples S1 and S2, coalescent to their most recent common ancestor at time T1, and then furthermore, their common ancestor coalesces with S3 to form the common ancestor of S1, S2, and S3, and so on. One of the key things about the coalescent is that the mutational and genealogical processes are decoupled. We can think about these things separately from each other. So we're going to follow that way of thinking today. So we're going to talk about the genealogies first, and then move on to the mutations afterwards. So from the beginnings of coalescent theory in the early 80s, simulation has been an essential tool. So coalescent theory gives us lots of analytical results, but some of our problems are too difficult to get exact solutions for. And so we've got lots of approximations, and simulation is often used to measure how good these approximations are. Another really important application of coalescent simulation, particularly today, I think, is generating null distributions. So when we have a lot of population structure, it can be very difficult to get analytical results, but we can easily do simulations to find a null distribution to see if chance alone can explain these interesting spikes in the data that we see. And oftentimes it actually is. The variance in the coalescent is massive, and this can explain a surprising number of things that we observe. Another thing people do with coalescent simulations is approximate Bayesian computation, inference of parameters. So what a simulation of the coalescent is it produces a single sample from a distribution of genealogies that are consistent with our sample configuration and the supplied demographic history.
What's sample configuration?
Well, sample configuration is basically your sample size and where they come from. So you've got 100 samples from Europe and 100 samples from Africa, that sort of stuff. We're interested in coalescent simulations here for this talk because the tree sequence data structure, which is the subject of the talk, emerges naturally from this coalescent process. Before we get on to the simulations, recombination is an important complication. And so very basically, the recombination takes two parental chromosomes and it crosses them over to form a single child chromosome. And the effect of that, as far as we're concerned, is that different genetic locations will now have different ancestries. We don't have a single tree which describes the ancestry of a whole chromosome. We've got a whole sequence of these trees. So this position here will have a different ancestry to this position here. And it turns out that this is quite a big complication. And the stochastic process underneath the coalescent recombination is often called the Ancestor recombination graph, which is a notoriously thorny and difficult object to work with. So exact simulation of this process for more than short sections and more than a handful of samples was considered completely intractable until very recently. Partly motivated by this inability to simulate the coalescent for long chromosomes, the sequentially Markov approximation was devised as a solution to this problem. So the SMC says that given a coalescent tree at a given genetic location, we only have to think about the immediately previous genealogy. So it only depends on the immediately previous genealogy in the sequence. And this is this is a big approximation, but it turns out to be surprisingly good. And using this approximation, we were suddenly able to simulate whole chromosomes, which is a big step forward. But even under this approximation, it turns out that simulating large sample sizes is prohibitively slow. So the tree sequence data structure is an efficient encoding of the correlated genealogies that we get under the process of recombination. And as I said before, it arises very naturally as a consequence of the simulation algorithm. And so to see how this works, we're going to do a step-by-step walkthrough of a particular instance of the simulation algorithm, so we can see how the structure arises and we can see how it's encoded.
So does the sequentially Markov, that doesn't play a role here, right? That assumption?
No, the SMC isn't used here yet. Okay, so the Coalescence simulation takes as input a set of chromosomes. So a set of sample chromosomes, which we show here, and they're all of length 10 in this case. So the top panel here shows the current algorithm state. This is all the stuff that the algorithm needs to know about. The goal of a coalescent simulation is to generate trees for every location in the genome. So we show the trees as they're being generated in this box down here. So we're at the present time, this is time 0, and all we know is that we have 4 leaves in our tree. So these are shown here. The tree sequence structure then is shown in this right-hand box over here. And this is the efficient encoding of the correlated trees that we'll be building up here. I'll explain how this works as we move along. So the first event that happens as we move backwards in time is a recombination event, which splits this lineage 2 here into 2 different pieces. So because we're going backwards in time, recombination has the effect of taking a single lineage and splitting it into 2 independent lineages at that breakpoint. And so that's what we see. We have two lineages now carrying this node 2, and we've got non-ancestral material over the bits where you didn't inherit it. The side effect of this event is that we now have 2 trees. Where we had 1 tree before, we now have 2, because the trees on either side of this recombination breakpoint can now evolve independently of each other. Moving on, the next event that we encounter is a common ancestor event. So we take these 2 lineages here and we merge their ancestral material into the common ancestor. And where we have overlapping ancestral material, we have a coalescence, and this means that these ancestors coalesce into a common ancestor over this particular interval. And so that's what we see. We now have a new node 4, which is the common ancestor of 2 and 3 over this interval 2 to 10. And this is reflected in the tree, which shows that 2 and 3, their parent is 4. So this information is recorded in our table of edge sets over here. And all this says is that from position 2 to position 10, the parent, or the 4 is the parent of 2 and 3. So we're just purely recording this tree node and the genomic interval over which it counts. So it's a very simple integer-based encoding of the genealogies. So moving on again, we have another common ancestor event, so between these 2 lineages, and it causes coalescence in 2 trees, so we update 2 trees at the same time, and this is reflected with 2 new edge sets now. So we have between 0 and 2, 5 is a parent of 1 and 3, and so on. We have another recombination event. This splits the tree which covered this interval into 2 trees, which now evolve independently. And the next event is a common ancestor. And we have another common ancestor, and again another common ancestor event until finally we have used up all of the ancestral material. We have complete trees at every position on the genome, and our simulation is finished. So these three trees, each over different intervals, are very highly correlated. They've got a lot of shared structure. And this shared structure is encoded precisely in this table of edge sets and nodes here. So just to be clear, a node here is one node that occurs in the tree. So node 5 here is recorded that happens at a particular time. That's shared across all of these trees. And an edge set then is just a particular assignment of ancestry over a particular interval. So 4, the parent of 2 and 3 is 4, which is shared between these two trees, and that's recorded in this one edge set.
So each of those trees is associated with an exact interval? Right?
Yes. Yeah.
Okay.
And the node times, just to make sure I've got it, are the time of the birth of a node.
So to speak.
Yes.
Exactly.
Okay. So we can write down this structure in a slightly different way, which shows how we recover the trees from it. So here we just write down the edge sets as the actual line intervals. So we draw a line for every interval, and we write the node assignment over it. And then we can see that finding the trees is really easy. If we want to find the tree at locus 5, then we just draw a line down through these intervals, and we apply the records as we see them. So here we've got 2 and 3 goes to 4, 1 and 4 goes to 5, and 0 and 5 go to 6. So a very, very straightforward algorithm for recovering the marginal trees given this structure. So MSPrime is an implementation of these ideas. It's a Python API which wraps a C library, and it uses HDF5 to store the generated tree sequences. So we just take these simple tables and we store them using HDF5's numerical storage routines. The Python API is used for running simulations, but it's also used for analyzing the data. So we've got a general purpose tree library here. And this allows us to analyze and generate the simulated data in the same script, which I think is a very powerful thing.
So what's the thing about new mutation in the context of this? Do you see what I mean?
New mutation?
Yeah, well, so you've defined an ancestral set of haplotypes that you're operating on.
Yeah.
But with the new mutation coming in, that will have a tendency to split things, and so are there parameters in that space?
These are just the trees. I mean, you can throw mutations down on this whatever way
at
you
any
want.
point.
Yeah.
So the argument is that the overall structure, and that's the balance between those two things.
Yeah. So it's completely independent of mutations at the moment. You know, when we throw mutations down on it, then the haplotypes will sort of just emerge.
So you can sprinkle them in.
You can totally sprinkle them in. Yeah.
In the tree.
So I throw a mutation down here, there, and then the haplotypes will sort of emerge out of that. So MSPrime is very efficient. I've shown here just a quick simulation benchmark comparing MSPrime to the fastest available Coalescent simulator, which is Scream. So what we've done here is we've, in the left-hand panel, we fix your sample size to 1,000, and then increase the size of the sequence we're simulating up to 100 megabases. And as expected, the time required to simulate using Scream increases linearly, right? Because it's using the SMC approximation. So what we see is that Scream, and we also show MSPrime here. So for less than 50 megabases, MSPrime is a bit quicker than Scream, and for more than 50 megabases, Scream is a bit faster. But it's worth repeating here that Scream is simulating the SMC approximation, whereas MSPrime is simulating the full coalescent recombination model, which was considered intractable. So what's more remarkable is when we look at what happens when we hold the sequence length fixed and then increase the sample size. And then we can see that the time required to simulate using Scream really just explodes. So there's no way we could simulate 100,000 samples using Scream. So in the interest of fairness here, I've shown two different results for MSPrime. One is where we output MSPrime's native HDF5 code, and the other where we output Newick trees, which is what Scream is doing. And we can see that the time required to output the Newick trees actually totally dominates the time required to run the simulation itself for sufficiently large sample size. So we have the structure that we've simulated. We'd like to analyze some of its properties. We'd like to find out how it performs at scale. And we can use coalescent theory to do this, which is a really nice property. So we've proved that the total space complexity of the tree sequence structure is order n plus rho log n, where rho is the population scale recombination rate, and rho log n is basically the expected number of recombination events. And then, alternatively, if we're going to use Newick, which basically just encodes every tree, and this would be the same for any tree format where you write out every tree without trying to capture the shared correlations. The storage space required is order n times rho log n. So they simulated 100,000 samples of 100 megabase human-like chromosome, and it took them 4 weeks using MAX, which is another SMC simulator. But using MSPrime, this only takes about 5 minutes and less than a gigabyte of RAM. If we were to output the results of this simulation as in Newick format, which is the standard, it would take about 3.5 terabytes of space, whereas using MSPrime, it takes about 90 megabytes. So we're about 40,000 times smaller in this example. Then if we want to actually use these trees, we need to read them in, we need to parse them to actually do calculations with it. And if we do this using MSPrime's Python API, it takes about 3 seconds to iterate over all 1.1 million trees. In contrast, if we use BioPython's Newick parser, which was the fastest one I could find, it takes about 3 seconds per tree. So this is about 1 million times faster to use MSPrime in this example. So there are big gains to be made. So I wanted to show a few applications of MSPrime pretty quickly. One application is benchmarking or variant processing software. So Ryan Lair and the others didn't actually care about trees when they did their simulation. All they wanted to do was to get some sequence data so that they could try out their GQT tool at scale and see how it performed. And because it took literally weeks to generate the data, they didn't systematically analyze the effects of different population processes.
I'm actually variant processing software for me is running on sequence data, and or BAM files really in practice, and you haven't got those. So what actually are you doing here?
We're taking VCFs and we're analyzing them.
Are you taking VCF as a starting?
No, so this is just about simulating it. We'll be operating VCFs definitely.
Okay, so I'm pressing you a little bit here. So you have your genotype data.
Yeah.
What's the software supposed to be doing?
Compressing the genotype data, making it more easy to deal with, and it's supposed to be processing the genotype data. So actually running calculations on it. So calculating allele frequencies, computing LD, all this.
Okay, fine. Well, thank you. I got it. Okay, very good.
Totally valid point though.
All right.
So in case you think that MSPrime can only be used for boring software development tools, I wanted to show an application, a real biological application that's been recently published from folks right here at the road. So Alicia Martin and others showed that the GWAS we perform on European bias cohorts, we need to be very careful about seeing whether those results port to other populations. A part of the study was a simulation study where we took 600,000 human samples of human chromosome 20, then built a sophisticated polygenic risk score model on top of this, and then examined the results. And what they showed is that across different random replicates, we see inconsistent and unpredictable biases arising between populations. So purely as a result of the stochasticity and the coalescence, we can see these very unpredictable biases occurring. And I really love this application for a few reasons. And the first reason, obviously, is that it's really important in science. People are doing really important science with MSPrime, which makes me very happy. But also, I think wouldn't have been feasible without MSPrime, this part of the study anyway. So simulating 600,000 samples wouldn't have been possible. But less obviously, I think, the simulations built on top of the data would have been an awful lot more difficult to do without the Python API. So all of the processing done with the data was done in a single script, and this involved no exporting of data formats, importing into different tools, exporting back to a different data format. It's all done in one script, which is so much easier. So yeah, given the genealogies, what can we do with them? And we can encode variation data on top of these genealogies. So just to recap on what the problem that we're trying to tackle is, present-day methods for storing and processing genetic variation data don't scale particularly well with sample size. And far from being a sort of abstract problem that we may need to deal with some way down the line, it is a very pressing problem that we need to tackle today. So we have very, very large data sets. We've got XAC with 60,000 exomes, and another project that I don't know how to pronounce with Nomad with
The GSA.
Okay, 120,000 exomes. And UK Biobank is just about to release genotype data for 500,000 samples. And all of these have millions of variants. And I think it's fair to say that working with these large-scale data sets is very difficult. And it seems likely that the data that we've produced at such great expense and great difficulty are not being exploited to their full potential. So the key abstraction underlying the methods that we have today for working with variation data is the variant matrix. Right? So we've got a set of samples and a set of variants. And if a particular sample carries a particular variant, it's got a 1. And if it doesn't, it's got a 0. So it's a very straightforward encoding. And yeah, I know I'm brushing a lot of details under the carpet here. What's also very straightforward to see is that if we have a million samples and a million variants, then this matrix is too big to work with. Because the space complexity is order n times n. People are very well aware of this, so we've got various different methods of encoding and compressing this matrix, which we're going to come back to in a bit. I'm going to be talking about the variant call format for quite a lot, so I think it's fair to explain what it is, even though everybody already knows this. So it's the standard method of interchanging variation data, and it's a straightforward text encoding of the variant matrix. So it's not a particularly efficient way of encoding the data, but it does compress pretty well. But it's quite poor when we want to actually process the data, because we have to decompress it, and then we have to parse it, which takes an awful long time. So the approach that I want to suggest is that we can encode this data instead using a genealogy. So suppose we know the genealogy. We have our samples 0 to 4, and our ancestors 5, 6, 7, and 8. And this is encoded using an oriented tree, which is very straightforward integer list encoding of a tree. So the parent of 0 is 5 and the parent of 1 is 5. So pi of 0 is 5, pi of 1 is 5. It's dead straightforward. And then we encode the variation that we see by putting mutations down on this tree. So we've got a variant here, and this corresponds to a mutation that happens over node 6. So 3 and 4 are 1, because these are the samples under that node, and everything else is 0. Again, we've got a mutation that happens at 5, so 0 and 1 are 1, and everyone else is 0. And we keep throwing down mutations on this tree. And the point here is that every row of this matrix is encoded by a single integer in this list mu. So now our space complexity is order n plus n, not n times n.
Are we assuming just a single mutation per tree?
We're assuming in this example here, because they're binary, we're assuming infinite sites. So we've got one mutation per site, but that's...
And do we also know what the ancestral allele is?
Yes, we are assuming that. There are reasonably pretty good methods for...
I'm well aware of that, but it's not perfect.
It's not perfect, fair enough. Yeah.
Okay.
That is the assumption we're making.
Okay.
Of course, we don't have a single tree. We've got a whole sequence of trees across the chromosome, as we were talking about a minute ago. But we know that we can encode the whole tree sequence in order n plus rho log n space. So therefore, the amount of space that we need to store the variant matrix for n mutations is n plus rho log n plus n. And this is still a huge difference to order n times n. So just to stamp home how much of a difference this is, I ran some simulations where we simulate human chromosome 1 with a standard demographic model with standard mutation rate, and did this for increasing sample sizes. So for 10 million samples, we have a file size of about 1.3 gigabytes. In contrast, the VCF for this is 318 terabytes, which is about 250,000 times bigger.
Did you actually make that?
No. I made 1/10,000 of it and then multiplied by 10,000.
You should do the squiggle. The squiggle equals.
Fair enough. Yeah. I thought it was near enough at this point. So there are huge gains to be made here, is the point. Okay, so the cost of very high compression ratios is nearly always a very high cost, corresponding cost in decompressing the data so you can actually use it. But contrary to this, the data that we store using genealogical encoding doesn't actually need any decompression because it's already exactly in the form that we need. So as a result, the information that we store is both smaller and faster to process than it is under other encodings.
You've glossed over a critical step, I think, which is you missed out the step of going from your variant data to the trees.
I've totally glossed over. That's the third section of the talk.
Okay, you're going to talk about that.
Yeah. This is still simulated data.
This is where you know the tree.
This is where you know the tree. This is a theoretical analysis at this point, right?
I'm throwing a mild rock or pebble.
Yeah.
When you say processing variant data, you're not really doing it.
Fair enough. Put a simulated in there.
Okay.
Yeah.
Okay, it's a totally valid point.
Or processing variant data where you already know the exact code.
Yeah.
Okay.
As I say,
That's doing the coalescence.
Yeah.
this is a theoretical analysis here. It is like, what could we do if we knew it?
Yes.
Yeah.
Is there still there must still be some more limited subset of operations that you can perform that are efficient? Is that, or?
Well, I think we need to explore that, to be honest.
Okay, so what, like minor allele frequencies on subsets and things like that?
Yeah, we'll look at a couple of benchmarks in a minute, but we need to systematically analyze this. There's a lot of work to be done.
Like every time you think of a new thing you want to do the data in an efficient way operating directly on this structure, you need to think of how to implement that.
You totally do that, yeah. So there's a lot of work to be done. Yeah.
Okay.
I've only, in terms of, you probably can do an awful lot of things directly on the tree if you have the tree available,
right?
But it does involve writing new algorithms for basically everything.
Right, because as soon as you're forced to sort of write it out to a matrix style data structure,
then you
lose everything.
You've lost, yeah, totally.
Yeah.
So I'm going to compare with some formats for storing and processing variant data. A lot of these developed here. So BCF is just a straightforward Z-lib compression of the binary encoded VCF. Plink uses an uncompressed binary format, so we expect that to be a bit bigger. GQT uses a clever succinct bit vector compression, which allows us to operate directly on the compressed representation for some operations. BGT from Hung Lee uses a pair of PBWTs to get really superb compression of the data. And Hale then, which I'm sure you're all familiar with, uses a distributed columnar format, which it allows it to sort of easily distribute, very good, easily distribute computations over a huge cluster. So it's a real win. So first of all, the file sizes. So again, this is simulated data. We simulated human chromosome 22 under a 3 population out of Africa model for increasing sample size, so up to 100,000 samples. And then for every tool, except MSPrime, we took this simulated tree sequence, converted it to VCF, and then imported that VCF into the tool in question, and then report the output file size. So as we expect, Plink is the largest here because it's uncompressed, and it's followed reasonably closely by Hail. GQT then seems to have pretty good compression. It's only slightly bigger than the BCF, and BGT wins the sort of compression contest by quite a margin here. So it's much smaller than the other tools. But MSPrime in the original simulation file is still substantially smaller than BGT. So we don't want to just store data, we want to run some actual calculations on it. So what we've shown here is calculating allele frequency within each of the 3 populations for every variant. And we output the time it takes to do that for all of these tools. So the first thing we might notice is that Hail is quite a lot slower than the other tools here, but this is because it's a very unfair benchmark to Hail, because we're measuring the total single-threaded CPU time, the total amount of electricity burnt, in effect, that it takes to run this calculation. And that's not what Hail is optimized for. Hail is optimized for running in parallel across a whole cluster of machines. And what you care about then is the elapsed wall clock time. So moving on, the next tool that we see is BGT. So what I think is happening with BGT is that it's paying the cost of its very high compression rate. It's having to work quite hard to get to run this particular simulation run this particular benchmark on the data. Plink is next, and then we see GQT here, which seems to be sort of straddling the middle ground between having really high compression ratios and being able to operate on the data efficiently. But then we see the MSPrime results, which are faster than all of the other tools, but more importantly, is growing at a much slower rate. So all of these tools grow at roughly a linear rate with sample size.
These subpopulations don't have to be adapted to the tree in any way?
No, it could be any arbitrary.
Any subset?
Any
It doesn't have
subset.
to be like clay?
No, absolutely nothing. And so these are all growing roughly linearly with sample size, but MSPrime is growing sublinearly with sample size. So we can see that as we go out here to 1 million or whatever, then we're still going to be able to run this benchmark in a reasonable amount of time. The next benchmark I'm going to show is computing LD. So compute LD genome-wide over 2 different window sizes using Plink and MSPrime. So what we see with small window size, there isn't much difference between Plink and MSPrime, they're more or less the same. But when we have a large window size, we see that MSPrime is quite a bit faster than Plink. And again, we see this property that Plink is growing linearly with sample size, whereas MSPrime is growing less than linearly with sample size. And this is a really critical property, I think. So to summarize this section, we talked about encoding variation data using genealogies. So suppose we knew what the genealogies were, what could we do in terms of encoding genetic variation data?
So in all these experiments, you simulated from MSPrime, and then you export it to all the formats.
Yeah, exactly. So what we showed is that we can potentially store data in a tiny fraction of the space, and we can also potentially run algorithms substantially faster than possible.
Is there a convenient way by which to associate individual specific information beyond simply the genotype in the tree structure?
That's an excellent question.
Thank you.
Yeah.
You want to repeat it for the audience so they can hear?
The question is there a way to associate individual data other than genotypes?
With each individual. So if I want to do a computation on every individual genotype by every, say, allelic depth for all genotypes, something we may be interested in doing from time to time, can we do it in this way? Because I think that's a very hard problem.
That is a hard problem. That is a hard problem.
Well, if the additional information is not adapted to the tree, then you're screwed. I mean, it's not going to be log n. So.
I agree. That's why I asked the question. I'm
And
curious about.
it is a question that is...
Information, theoretically, it's not possible.
It's a hard question. I don't know the answer. Is the short answer.
Okay.
We're not far enough down this path.
I see.
So.
All right, that's fine.
Adherence to everything is that stasis will not be a big fan. In this audience,
Yeah. So what I would say, I suppose, is that this is a problem that any method will have to deal with.
Right. Agreed.
Right? So any method that's good can be adapted into this framework. I don't know if there's a...
Yeah, I mean, I think it's a reasonable stance to say for some more complicated kinds of analyses where you want to do it very, very rapidly and iteratively, that getting to a genotype-only product and then compressing it in the clever way that you do so is a very sensible future direction.
Yeah.
But I think we do also need to have an ecosystem of tools for the different purposes.
Sure.
Because we do want to do like QC and
Yeah, of course.
all that kind of stuff.
Yeah. I think this is ideally for the end point, the end result of all the QC.
I'm sympathetic to working on that end.
Yeah.
Like, being able to download the genotypes for UK Biobank on a laptop, now everybody can process that data without having a compute cluster. That's pretty cool.
Well, they could if we could.
Right, I mean, assuming it's public and
Well,
tree structure,
there's a bigger problem to be solved.
We've all been
Okay, so, all right, assuming we could do this would be brilliant, right? That is a very, very, very big if, right?
Is it easier to do a probabilistic extension instead of having our
That's the other question.
distribution? Because that's like UK Biobank has that data already.
Well, if you can map it onto the tree, then yeah. Right? If you could map your probabilistic model onto the tree, then, or you could, I mean, there's a lot of times the tree will be unresolved in places, and you can just use this probabilistic data to help you resolve the tree. You know? I think it's a hard question to answer because we just don't know. You know, there's.
I think you can probably going to be correct. It's just errors in the data. There's back mutations a lot.
Yeah.
These probabilities have no tree structure. This is great. Well, you're getting
So, yeah, I vote for let John go.
I agree with that. I want to see this part. This is the part I want to see.
You're certainly heightening tension effect. Good narrative.
Yeah.
Structure. And that's it.
Thanks for the
We're done. Okay, so the third section of the talk, and by far the shortest section of the talk because it's ongoing work, is talking about how we estimate these trees from observed data. There's a very rich literature on estimating trees, or a tree, for a short section of sequence data in which there isn't recombination. This is basically phylogenetics. But estimating trees in the presence of recombination is a much more difficult problem and hasn't been investigated to near as much depth. There has been some recent work on estimating trees genome-wide, things like Argweaver, but nothing on remotely like the scale that we need to tackle the problems that we're interested in tackling. So just to state the problem again, we want to estimate a tree sequence from observed variation data. So given a VCF, try to estimate the tree sequence that's underneath that. And we know immediately that trying to fit any complex model to this is going to be intractable. So we need some sort of simple approximation that we can scale. So
if I could just put in a quick comment. For your coding problem, you do not really want to estimate the tree. You just need a tree, a good tree that works.
Yeah, totally.
It doesn't have to be a statistically good estimate.
Yeah, absolutely.
Okay,
just needs to be consistent with the data.
Yeah.
You might argue that a good tree would provide good compression.
Yeah.
So there might be a middle ground in between the two. Scale is our first property that we must have, and second property we must have is it must be robust to incur. You know, there are errors in the data, and we need to be able to deal with it. So I'm going to give a very quick outline of the inference algorithm that we're working on. don't have time to go into the details now, but I'm more than happy to go into the details afterwards if anyone's interested. So it's got 3 basic steps. We first infer a set of ancestors based on their relative allele age, and then we take these inferred ancestors and we run Lee and Stevens copying model on that. And so we set up a copying process amongst the ancestors and then copy from the samples back into the ancestors. And then the result of this large copying process is a big matrix, and this matrix has genealogies embedded within it. So we can take this matrix with embedded genealogies and convert it into an MSPrime tree sequence using some fairly straightforward algorithms. So that's the rough outline of the algorithm. So we want to check, do some basic sanity checks on our algorithm to see if it has some of the properties that we need of a tree inference algorithm. So we want our trees to be reasonably close to the truth. We don't need them to be particularly good, but we want to know they're not completely bad either. So the way we do this is by comparing the estimated trees. So we run some simulations, we have the trees, we know the truth, and we run our inference algorithm on some mutations that were thrown down in those trees, and then we measure the distance using tree distance metrics from the inferred trees back to the original trees, and the closer we are to 0, the better, right?
But as Nick said, if you don't really care about the population genetics underlying your... In principle, is this necessary?
Well, I think it's a good idea to make sure it's not completely wrong, right? If it's getting completely mad trees, then it's very unlikely that it's going to do good compression. Right, so as we increase the mutation rate then, as we get more and more information about the topologies of the trees, what we should see is that the distance from the estimated trees to the true trees is trending towards 0. And again, being robust to a reasonable amount of error is absolutely essential here. We need to know that we're doing well in the face of errors. So what we've done here is we've simulated a short chunk of human-like data for 50 samples, and we've done this for 3 different error rates. So no error, 1% error, where we take some of the mutations that we've thrown down, we flip them, we throw some errors down onto the data, and then a very large amount of error. And so what we see is that the general trend for all of the methods is that as we increase the mutation rate, the distance back to the true trees is trending down towards 0, as we'd expect, so that's all good. Yeah. So then in the error-free case, our tool TS-infer is doing surprisingly well. Right, so we're roughly the same accuracy as Argweaver, which is a very heavy-duty probabilistic model. And then, very encouragingly, as we throw more errors on, we're actually doing pretty well as well. So for the sort of intermediate errors regime, which is kind of where we expect to be, we're pretty much the same accuracy as Argweaver, which is very encouraging.
So the error rate here is static for the number, is a static fraction of the variant data you're looking at?
Yes.
Yes. So that upward slope of the back end of the 10 to the minus 6 for a high error rate?
That wouldn't be good, no.
That's a little worrying, isn't
okay.
it? Because you're not showing consistency with respect to the... It's almost like the number of errors is mattering more than the fraction of errors, if you see what I mean.
Okay.
Because with a higher mutation rate, you have more variance,
yeah.
and then you're also going to have more errors, and so your tree structure is going to be more garbage.
But this is a lot of error, right?
I agree.
Yeah.
I agree.
So hopefully...
If we're doing that badly, nothing's working anyway, so it's sort of moot.
Yeah, I think so. Well, we're hoping that this is reasonably encouraging. Anyway.
And is the error just a site that's not real, or a genotype flip in a site that is real? Because that will make a very, very big difference.
So the model for errors is to flip bits, but to make sure that we maintain the overall allele frequency in a particular site.
Right, so you're not innovating new sites.
No.
Explore that.
Okay.
Because I think that's going to have a lot of problems.
Okay.
Because that will create a shattering of your tree in a sense. And I think that, and that's a more realistic error mode in some ways.
Okay.
So screwed up genotypes is definitely part of it, but
Yeah.
sites that are not real sites, but are actually errors that we don't really control properly.
So a low frequency site, a low frequency variant that is not real, is it?
Correct.
Okay.
That happens.
Yeah, totally.
True story.
Yeah. We'll definitely look into that, thanks.
Have you explored whether it makes any sense to build trees from all variants above a certain threshold?
Yeah, I was
rare
thinking the
ones
same
on
thing.
later? Or
Maybe.
how different do your answers get? 'Cause you probably get the layout of the recombination with much cleaner data.
Yeah.
Yeah.
I guess what we're actually no, I think we're using that information because we're starting with the oldest ancestor, and that's the most frequent, right? So the sort of base structure is being determined by the oldest mutations, which we hope are the most robust to error. And then as you come up further, so singletons don't affect the tree structure at all.
You're kind of ordering by allele frequencies.
Yeah. I can take you through the algorithm in a
Yeah, that'd be
minute.
great for the discussion. We
Yeah.
should finish up fairly soon. The usual format is that we then have another half hour to talk.
Okay, cool.
Yeah.
Well, we can go through the details and I'd be more than happy to do it.
Yeah, we can do that.
Okay. So the take home here is that it's a very small region, and it's a proof of concept implementation of the algorithm, but it's reasonably encouraging. We're moving in the right direction. So yeah, to summarize this section, we've got an approximate method based on the Lee and Stevens copying model. Performs reasonably well on small scales, seems to be robust to certain types of error, which may not be indicative. Our initial proof of concept doesn't scale, and the compression results we have are unspectacular, but I'm pretty sure I understand why, and we're working on a second iteration of the algorithm. So basically it's a work in progress, and we hope to have some good results to show pretty soon.
You say it's doing well in terms of accuracy. What about speed compared to the other tools?
Yeah, I mean, it's a proof of concept. I think it's a bit quicker than Argweaver, but that's about it. It's not fast, but we didn't try to make it fast.
I'd like to make a very emerging comment. I'd like to make a comment here.
Yeah.
You can really afford to do quite a lot of work at this tree determining stage. I mean, the reason is there's a whole lot of computations behind all this, including aligning all your sequences that's been done.
Yeah.
And this is on a given data set. This is just a single. You're finding a compression algorithm, which you can decompress very fast.
Yeah.
And you only have to do it once.
Yeah.
You have your million samples. They come in. You do this thing.
Yeah.
When you've already had to do all the alignment, you can actually stand quite a lot of work here.
Yeah.
Huh?
I completely agree. If we can get something that we can scale reasonably well and parallelize, then I think it works. So just to finish up, I don't want to give the impression that this is completed work, or you can run all of your analyses using this software. There's a lot of work to be done. Obviously, the biggest open problem is converting real data into the format. But given that, I think there's a lot of interesting computer science problems to tackle. For example, if we knew an efficient most recent common ancestor algorithm that worked across the tree sequence, this would have a lot of different applications. And we have to wonder that if, given again that we can estimate this structure, does this give us a new insight into more biologically motivated algorithms, such as phasing, chromosome painting, and stuff like that? So I think these are really nice open problems. So that I've just unfinished. Thanking my collaborators and letting you know all the different places you can find MSPrime and where